whole speech. Sorry, guys. But thanks for joining us again. I just want to say I'm Jenny Chen, founder of 3D Heals, and about the panel. Number 2 mission is help creating a platform, whatever format it is, could be in person or virtual, to help you guys connect with one another, to form companies, collaborate, partnerships. Give him a shout out, and he'll meet up with you maybe someday in person. So Greg, I want to give the podium to you.
As biomaterial development progresses, it drives the expansion of 3D printing. I reiterate, biomaterial development drives the expansion of 3D printing and the industry capabilities, which allows for new solutions for personalized healthcare and revolutionizing patient care and treatment outcomes. Very good. Gerker,
awesome.
Say hello just to make sure that your audio is okay.
Yep. Hello. Thanks for the introduction, Craig, and thanks for joining us, everyone. I'm excited to be here. 3D Biofiber. 3D Biofiber, just as a high level, is a protein fiber manufacturing company. So we're a company focused on making protein fibers accessible in tissue engineering, and a specific focus of ours is on 3D bioprinting, which I'll focus on today. Mechanical durability. And collagen fibers are the most abundant protein structure in the human body. They naturally play a key role in maintaining the structure of our tissues, and they're highly physiologically relevant in terms of their definition in the extracellular matrix. The way that I explain the process is, if you think about a spider spinning a web, which is another example of a protein fiber, it involves a spider crawling across the ceiling, depositing some solution, and dropping itself down. Production. Our technique at 3D Biofibers make collagen fibers that match or exceed their strength in the body, where their main role is to provide structural support to tissues. Printed scaffold that is reinforced by our fibers, improving that mechanical durability over time. And this is a project that we've been working on that's been quite successful. So I'll go over some of the high-level data about how we're really solving this challenge for the industry. The first is the mechanical durability. When I started this, I was saying the main challenge is that constructs lose their shape over time. In this example, we've used on the left side here a construct of gelatin methacrylate, commonly called GELMA, which is a very common bioink that's used by the industry. And in this left image, we've seeded it with fibroblasts, just a type of cell, and you'll see that within 6 days, 92% of the shape. So we're really solving that mechanical durability challenge, which allows us to keep over 90% of these cells alive throughout this time, really driving that ability to grow these constructs to reach their full maturity, commonly known as shear thinning. In this example, we have 5 conditions. The black lines at the bottom is our GELMA-only control, which operate. That with our fibers, that rheology, viscosity, and printability is essentially completely maintained. Whereas when you use other additives, it can be increased by 2 or even 4 times, really making it difficult for end users to integrate that without having to redefine their entire printing protocols. And then when we look at the load, so the overall strength, whereas when we add our fibers, you'll notice that those cells are much more elongated. They're proliferating. That's why they're clusters of these cells that have replicated out, really driving their functionality. And very importantly, it allows people to decouple their local construct stiffness from their overall durability. So you can not only have a construct that's durable, but you can also create a mechanical environment that is suitable for the specific cell type and application you're working on. And we're able to do this by creating an additive that's really representative.
For those of you that have questions, feel free to enter them in the Q&A box. I'll start off. Could you speak to the consistency from batch to batch? Because I know that's usually a concern in this field. What are your tactics for being able to raise so much funding in a short period of time and be able to?
Yeah, so a big part of it is baked into the business model. So at the core of our business is this technology that is the ability to make collagen fibers at scale. And collagen fibers, being the most abundant protein structure in the human body, have a very wide range of applications across tissue engineering.
There we go.
Hello.
All right, very good. So Julian, let me give you an introduction, and you can pull up your slides in the interim.
With already validated material, so injectable material, the one that the industrial currently use. basically depending on the material you use, but in the case of silicone, you just have to cross-link the material and remove it from the powder. And the beauty of the process is that you can reuse the powder for the next printing. So the support you use, and in terms of material compatibility, what we have been able to prove the last 3 years is that from show 1 to show 80. So how do now we apply this process in healthcare? So basically with using silicone-based material, the idea was to develop soft medical devices. So the ability to mimic soft tissue. And for this, so personalized medicine, patient-specific implant, surgical model to train the surgeon. So as an alternative for cadavers of animal experiment, we can really, and of course, which is also a improvement for the surgeon, the material that we use can be. So now it's a brilliant example of how you can combine different 3D printing techniques. As we can work with already compliant material, the next step for us is to develop really implantable medical devices. An hospital in France. So they came with their idea, and then in 3 months, we were able to develop the first working prototype with our techniques. And now we are at the stage when we were able to develop this patient-specific class 3 medical devices.
Might influence the print speed.
Yeah. Now, like we just maybe compare to maybe what people know about silicone-based extrusion technique. Most of the time, silicone is highly viscoelastic material. When you increase too much the speed, basically the filament will just stick to the nozzle, you know, too viscoelastic actually. And so the speed is always limited to, I would say, like 10 to 15 millimeters per second when you print. But in our process, as we can basically trap the filament inside 2 powder layers.
I believe you're on mute, so feel free to unmute yourself. Yes.
Okay.
So why don't I introduce you both, and then Needy, you can take it away with the presentation, if that works.
Sure. Yeah.
Okay, very good. So Needy, to start with you, I think seems like you're quite involved in the organization. If I am understanding correctly, you're the director of R&D, business development, and quality affairs. So it seems like you wear quite a few hats, and I'm sure we'll learn a little bit more about a day in the life in your presentation. Dr. Biswada, as the scientist and entrepreneur, you found the company in 2003. We'll certainly let you speak to that, or Needy speak to that during the presentation. I was quite impressed reading your background that you also have experience for over 20 years in Ethicon with J&J, doing a number of years of research and development. So both R&D and entrepreneur experience.
The strength and shortcomings. Today, we are only going to focus on synthetic bioinks because in many cases, these synthetic ones helps overcome the challenges observed with natural bioinks in context of achieving certain target product requirements being marketed by Abirex. These products are based on our absorbable polyurethane technology platform, and they are very safe and biocompatible. And we also have received NSF phase 1 and phase 2 grant for development of polyurethane-based internal tissue adhesive. In addition, we also do custom synthesis and bulk scale manufacturing of drug delivery polymers. And over the last 5 years, we have ventured into the area of 3D bioprinting, wherein we are working with 3D printing companies to design and develop custom bioinks for various applications. So this is our journey. Let's go to the next slide. So we have an extensive experience in developing synthetic absorbable polymers that are very safe and biocompatible deposition. And one of the important ones is they need to have mechanical properties that can match the intended tissue or organ that we are printing it for. And they should allow even distribution and cell viability within the ink that can support the cell growth, differentiation, and proliferation, synthetic ones. And then there's another class of polymers that are totally synthetic, which you can see in the last 2 boxes. They can be PEG-based, PDS and PCL, polydaxanone and polycaprolactone polymers are widely used for 3D printing applications. There are PLGA polymers, polyester amides. And the second class of polymer is PEG-based. We have combined the technology where we use PEG and combined it with our biomaterials, which is very safe and biocompatible, and come up with a new ink for organ bioprinting compared to PLA. So if you are looking for little different properties, but strength, and you want faster absorption than PLA, we can go with PDLA polymers properties. So as I was mentioning to you in my presentation earlier that we are going to cover our two technology platform very briefly. So this is one of our unique technology. Hey, I would like to thank you all for your time, and we are happy to answer if you have any questions. We are here.
It's really a good one, specifically exploring hybrid bioinks, a combination of both natural and synthetic polymers. Harry Fu is interested if this is something that you've already explored, and if yes, do you find them beneficial? Well, thank you both. And again, Biswata Biomedical, to reiterate, they sponsored today's event, so we want to thank them as well. I shared their information in the discussion, and I would encourage everyone to read up more about the exciting and interesting research that they're presenting. Health. Nicole grew up in Michigan before moving to Boston to attend Boston University. And of course, she's in Boston, as we just said today. She studied biomedical engineering, and she did have a minor in mechanical engineering.
Thanks, Greg. Yeah.
So without further ado, Nicole, why don't you take it away?
And our current problem right now that we are trying to solve is that of chronic eardrum perforations. And if those of you don't know what your eardrum is, it's essentially a thin tissue that captures sound waves from the environment and conducts them into the ossicles in your middle ear. Your eardrum has this very carefully aligned circular and radial collagen structure in it, which you can see in this SEM image that actually contributes to this ability to conduct sound. Medial side of the eardrum. Unfortunately, these autologous tissue grafts are imperfect. They have varying thicknesses, and they importantly also don't match this structure. They can also be very difficult to harvest in place. And so what you're essentially doing is just creating a bandage on your eardrum with this piece of tissue at high frequencies. And furthermore, after these grafts are implanted, we see remodeling of these grafts into tissue that matches that microstructure, which I'll talk about. And so this is just a quick animation showing how these grafts are printed with different support layers in order to achieve this bilayer architecture, which you see here. We've done some pilot studies with these grafts in chinchillas and shown that not only are they quicker to place than the predicate device, but they also are rated as more secure by the surgeon. So overall, we're hoping that these designs really, open up the market for tympanoplasty and encourage more people to undergo this procedure and get it fixed in a less invasive manner. So this includes children, especially those whose ear tubes have fallen out, to get it repaired quicker, elderly patients that often can't undergo general anesthesia, patients in rural settings, and also patients in developing countries. And so where do we go from here? Beyond the phonograph, our team is interested in partnering with medical device companies and materials companies to see where we can move the nozzle in healthcare. And so we had recently released this new add-on for the 3D bioplotter called the print roll system, targeted at cylindrical conduits, so vascular grafts, nerve conduits, and things like that. And so particularly partners that are interested in those space would love to talk to you.
So why don't we have all of our panelists unmute themselves, and we can conclude the discussion by being able to open up the floor.
Synthetic reproducibility is always good. I've been working on medical devices for years. That's the reason we took as a challenge for us to start working on synthetic materials. 50 years ago,
in healthcare that I would never be able to learn about and be excited about. So it's a very enjoyable process for me, and I'm very appreciative of all the speakers who have donated their time today to join us.
we will definitely come.
Yeah, stay tuned. We'll make some announcement to figure out where we're going to be. So we're going to log out of this webinar, and then when you go online back to the Zoom event hub, go to the top bar, it says Expo. Go to the Expo and then go to our sponsor booth, which is Biswata Biomedical. Click on that, enter the booth, and then enter the meeting in the booth. And this is how we can see you in person. Risk is low, return is high. Again, I want to thank everybody for joining us today, especially Craig for doing such a wonderful job moderating. You did a much better job than I could have done.
You've done a great job, Craig. Great job.
Yeah,